In this video, I'm going to look at antibody production and the recombination of the genes that produce the antibody. So first of all, let's look at the structure of an antibody. An antibody is made up of 4 chains of proteins linked together. These 2 chains are called the heavy chains. And they're linked together. Then there are 2 other chains linked to the heavy chains, and they're called the light chains because they're smaller. And these light chains are linked to the heavy chains. And that's what gives the antibody its Y shape. Now there are some regions that are very important on the heavy chain and the light chain. These regions are what define the style of antibody. Now the most important region are on the end, which I've shaded in here. And they're on the one on the end of every heavy chain and one on the end of every light chain. And they're called the variable region, which is what gives the antibody its specificity for a pathogen. So they're those shaded in ones there. The rest of them are called the constant regions, and they're not shaded in. And these are what define the style of antibody. For remembering, we also call an antibody an immunoglobulin, or just Ig for short. Now there are 5 isotypes, or just different styles, of antibody. And they're known as IgM, IgD, IgE, IgG, and IgA. This means immunoglobulin M, immunoglobulin E, etc. Now just keep those in mind. We'll explain what they mean later on. So what basically happens is that a progenitor B cell, which will give rise to a B cell, has a bunch of DNA inside it for making these antibodies. Now to get some variability, it shuffles up the DNA before becoming a B cell, so each B cell will have different antibodies. And these are pretty random. But remember that one B cell will produce only one type of antibody. So what we're going to have a look at is how a heavy chain is formed. And this is a heavy chain recombination of the genes during the DNA shuffle. And it happens in a kind of organized way. So we're looking at the germline DNA. So this is the original DNA. And there's a whole long string of DNA with various regions on it. And I'll draw it out schematically here. So imagine this is a long string of DNA with various genes on it. This region is called the V region for variable, this one the D region for diversity, the J region for joining, and then an M, a D, a G, an E, and an A region. And these regions are the constant regions. You might already notice that they're the types of isotype we talked about before. Now there are 40 parts to the V region. There are 25 parts to the D region, 6 parts to the J region. Now initially, one part of each of the D and the J regions gets selected and the rest get cut away. So we're left with just one part of the D region sitting right next to one part of the J region. And we call this the DJ recombination. The V region is still sitting over here like this, and the constant regions are still sitting on the end like this. So this is the DJ region, this is the V region. And the M, D, G, E, and A regions are still sitting on the end. Now the next thing to happen is that one part of the V region gets selected and the rest cut away so that it sits right next to the DJ region. This is what we call VDJ recombination. So the M, D, G, E, and A still sit on the end, but the V, D, and J sit right next together, like this. So this is what we call the recombined DNA. And depending on which part is selected, many different types can be created. Now this whole piece of DNA gets transcribed, so it becomes a string of mRNA. Sorry, I didn't mean to put protein there, I'll change that in a moment. So here's a V, D, and J region, with still all the constant regions on the end. But this is a string of mRNA now. Now one of these regions, for example, let's use the G region, gets kept while the others get spliced away. So that's mRNA. And this gives us a new spliced version of the mRNA with the V, D, and J region sitting right next to the constant region. So this is a V, D, J, and a G mRNA. Now that will get translated into a protein. And this protein will be the heavy chain of the antibody. Now this part will be the constant region, which is the G region, and the bit at the end will be the V, D, and J region. So this is what we call the IgG heavy chain And it's an IgG style because we chose the G constant region in our splicing. Now a very similar thing happens with the light chain recombination, except that there's no D region, so it's just a VJ recombination. But the rest is exactly the same. And that'll give us the two parts to make up our antibody. And that's an overview of antibody production.